You know, it's kind of crazy when you think about it. Like, when it comes to breast cancer treatment, you'd think we'd have everything figured out by now.
Yeah.
Especially, like, the really important things, but there's this one key factor, this thing called Ki-67, that's been surprisingly tough to get a consistent read on.
Hmm.
And it's huge GE, right? Because it basically tells doctors how fast those cancer cells are multiplying, which is super important for deciding, the best way to treat it.
Oh, absolutely.
Mm.
If you've got variability in how you're assessing that Chi-67, well, that has a direct impact on the treatment decisions that are made for patients.
Exactly. And that's exactly what we're diving deep into today. We've got this super interesting research paper that's all about a brand new approach to this whole Chi-67 thing using AI
Yeah.
to get a much more precise measurement, like right down to the individual cells in those tissue samples.
Wow, that's incredible.
Yeah, and this isn't just like some cool tech thing. This is about getting a really clear picture of how your cancer is behaving, which in turn can lead to way more informed and targeted treatments.
Yeah, I mean, when you think about AI in medicine, it really has the potential to bring this level of standardization and objectivity that, we haven't always had.
For sure.
And this research really dives into how a specific type of AI system, a system called YOLOv8, can be used to measure this really critical marker, Chi-67,
YOLOv8.
hopefully taking care of some of the inconsistencies we see with the current methods.
So our mission, if you will, is to break it all down. Like, how does this AI actually work, and why could it be so much better than what's being done right now to figure out Chi-67?
I think that's a great mission to have. But before we get into the AI part, I think it's worth just taking a step back and remembering how important Chi-67 really is in the whole scheme of breast cancer.
Totally.
I mean, we all know it's the most common cancer affecting women worldwide, sadly.
Unfortunately, yes.
And over the years, the way we, look at and understand breast cancer has become so much more, how do you say, nuanced.
Refined.
Yes, refined. Doctors now categorize it into these main molecular subtypes, right?
Right.
Luminal A,
Luminal B,
HER2, enriched.
and triple negative.
And Chi-67 really comes into play. It really matters when we're talking about luminal A and luminal B.
Okay.
These are the subtypes that are fueled by hormones, what we call hormone receptor positive.
Gotcha.
And what makes them different, what really influences the course of treatment, is how fast those cancer cells are dividing.
And that's where our friend Chi-67 comes in.
Exactly. The Chi-67 index measures just that, the proliferation rate.
Okay, so let's break this down for a second, because I
Yeah.
think this is where it can get a little confusing for people. So you have a luminal A tumor, right?
Right.
And it's got a low Chi-67 index, which basically means it's growing slowly.
Mm-hmm.
Doctors might only treat it with endocrine therapy, which blocks those hormones that are feeding the cancer.
Exactly, because in those cases, those tumors tend to be, less aggressive. They're not growing as rapidly.
But then on the flip side, you've got a luminal B tumor.
Yeah.
And if that one's got a high Chi-67 index, meaning it's growing more quickly,
More aggressively.
then it's a different story.
Yeah,
They might use chemo on top of the hormone therapy to really knock it back.
exactly, because we need to hit it hard. It's not enough just to block the hormones in those situations.
So it's not just about knowing the subtype.
Right.
It's about understanding how aggressive it is within that subtype.
Precisely. And that's why this Chi-67 index is so valuable.
And this isn't just something doctors kind of eyeball, right? Like, it's actually part of the official guidelines for treating breast cancer around the world.
Oh, absolutely. The importance of Chi-67 is well established.
Okay.
You've got the 2021 St. Gallen Vienna Consensus Conference. You've got recommendations from the International Chi-67 and Breast Cancer Working Group. They all highlight how useful it is clinically.
Yep.
I mean, they've even suggested specific thresholds. So, for example, if your Chi-67 level is below 5%, that might mean you can avoid chemo altogether.
Yeah, wow. That's big.
But if it's 30% or higher, then that really points to the need for chemo.
So those cutoffs are pretty serious stuff. Getting that Chi-67 number right is obviously a huge deal.
It's paramount for the
Yeah.
clinician and, more importantly, for you as the patient.
Yeah, totally. And I remember reading that it's even used in some of those really advanced genetic tests like Oncotype DX.
Yeah, the one that helps predict the risk of the cancer coming back.
Exactly.
Yeah, that just highlights how well established it is as a tool to help us understand, the prognosis and make those really important treatment decisions.
Okay, so we know how crucial Chi-67 is. But here's the thing, and this is where it gets kind of interesting. The traditional ways of actually measuring it, well, they sound kind of limited. Like they
Yeah.
literally involve looking at stained tissue slides under a microscope,
Yeah.
right?
Yeah, so the conventional methods, the ones that have been used for quite some time, they usually involve a pathologist visually examining the stained tissue.
Hmm.
And sometimes they manually count the number of cells that light up with that Ki-67 protein.
And all that visual assessment, I mean, that sounds like it's pretty open to interpretation.
Yeah, it's subjective. You have different pathologists. They might look at the same sample,
Yeah.
and they might come up with slightly different Ki-67 scores.
Right, which is that whole interobserver variability thing.
Exactly.
And that can lead to some pretty big inconsistencies in the results, which is obviously not ideal.
And not ideal for you as the patient.
Right, exactly. And the manual counting, oh my gosh, that sounds so tedious. The paper said it can involve looking at anywhere from 500 to 1,000 cells, like tiny dots, one by one.
I mean, you can imagine staring at all of those cells.
Oh my gosh.
It's very labor-intensive for the pathologist.
Yeah.
And even when they're being very meticulous, there's still the chance for those tiny little subjective biases to creep in, right?
Totally.
Just imagine looking at hundreds and hundreds of tiny cells under a microscope. You can get tired. Your eyes might play tricks on you. And even just a slight difference in how someone interprets, a partially stained cell can make a difference in that final count.
Oh, yeah, for sure. And when you think about what that means in real life, it's kind of scary because even a small variation in that Ki-67 score can mean the difference between getting chemo or not.
Absolutely. That's a life-changing decision based on a number that might not be as accurate as we'd like it to be.
That's a great point. So clearly, finding a more objective and consistent way to measure Ki-67 is really important.
Incredibly important.
And that's where this research comes in with its AI-powered solution. So this paper talks about using an AI system called YOLOv8, which is an object detection framework. And for those of us who aren't AI experts, can you explain what that actually means in simple terms?
So imagine YOLOv8 is this really, really smart computer program, right?
Okay.
And it's been trained to look at an image, like a picture, and pick out specific things in that image.
Okay.
And not only does it tell you what those things are, but it also draws a little box around them, like a digital box.
Oh, interesting.
And that box is called a bounding box.
A bounding box.
And in this case, the things that YOLOv8 is learning to find, they're not cars or people, they're actually Ki-67 positive and Ki-67 negative tumor cells
Gotcha.
in those digital images of those stained tissue samples we were just talking about.
Wow, so instead of a person having to look at it and guess, this AI is doing the work of finding and sorting those cells automatically.
Exactly. And the study used high-resolution images too, like really zoomed in at 40x magnification.
Wow.
And they focused on specific areas called Ki-67 hotspot regions.
Okay, what are hotspot regions and why are they so important for this?
So, Ki-67 isn't always like evenly spread out in a tumor. You'll have these areas where there's a much higher concentration of those rapidly dividing Ki-67 positive cells, and those are the hotspots.
So they're like the areas where the cancer is really active.
Exactly. And those regions are generally considered to be the most representative of how aggressive the tumor is overall.
Gotcha.
So pathologists are already trained to find those hotspots as part of the standard Ki-67 assessment. And in this study, the AI is trained to focus on those same areas as well.
Okay, that makes sense. So you're basically zeroing in on the areas that matter the most. And they looked at 30 cases of invasive ductal carcinoma, right?
Right.
So how did they actually go from those physical tissue samples to the digital images that the AI can work with?
Okay, so it all starts with standard clinical procedures. First, they take those little pieces of tissue, from biopsies or surgeries, and they preserve them in these blocks called formalin-fixed tissue blocks.
Mm-hmm.
And, of course, they got ethical approval to do all of this. It's really important to do things the right way.
Absolutely.
And then they do something called H&E staining, which is a really common technique in pathology. Yeah, hematoxylin and eosin. It basically helps pathologists see the overall structure of the tissue and pick out the specific areas they want to look at more closely for Ki-67.
Oh, I see.
And then they use another special staining technique using a Ki-67 antibody.
Oh, okay.
And that makes the Ki-67 protein visible. So the cells that are positive for Ki-67, they'll have these brown nuclei while the background is blue.
So the brown's like the signal that the AI is looking for, right?
Exactly. And get this, for each of those 30 cases, they took 6 to 7 pictures, which is a lot.
Wow, that's a ton of images. Why so many from each case?
It's all about getting a really complete picture. Even within those hotspots, there can be some variation in Ki-67 expression.
Ah.
So by taking multiple images across the area, they can get a more accurate understanding of the overall Ki-67 activity in that sample.
So it's like taking multiple samples to make sure you're not missing anything, right?
Exactly. So now the AI has all these digital images, but how does it actually learn to tell the difference between a positive cell and a negative cell? That's where the training comes in, right?
Right.
This is where it gets really cool. So they have this expert pathologist, someone who's really good at recognizing these cells.
Mm-hmm.
And this expert goes through all 180 images and manually labels each cell.
Wow.
They draw these little boxes, those bounding boxes, around each cell. Red boxes for positive cells and green boxes for negative cells.
Oh, like color coding them.
Exactly.
Smart.
And they use a special software called Label Me to do this.
Oh, okay.
And this meticulous labeling, it creates what we call the ground truth. So it's like the answer key that the AI will learn from.
Teaching the AI, this is what a positive cell looks like, this is what a negative cell looks like.
And the study also mentions this thing called an IOU threshold of 0.5.
Oh.
Yeah, it stands for intersection over union. And it's a way to measure how much the AI's prediction, that bounding box it draws, overlaps with the box drawn by the expert.
Oh, okay.
And 0.5 means that at least 50% of the AI's box has to overlap with the expert's box for it to be counted as correct.
So it's like they have to agree at least halfway.
Exactly.
Yeah.
And this is especially important when you have cells that are bunched together, maybe even overlapping a little bit.
Yeah.
It helps make sure that the AI is actually identifying each cell correctly and not just highlighting a whole area.
So it's all about precision. Okay, so they've got the images, they've got the AI, they've labeled everything. What else do they do to make sure the AI is as good as it can be?
Well, they use this really cool technique called data augmentation.
Data augmentation, what's that?
So basically, they take the original images and they create slightly altered versions of them.
How do they alter them?
Like they might resize them a little, rotate them, flip them, adjust the brightness, or even crop out little sections.
Why do they do that?
It's all about making the AI smarter, more adaptable.
Okay.
You see, by showing the AI the same cells, but under slightly different conditions, it prevents the AI from just memorizing the exact images from the training set.
Oh, I see.
It helps it learn to recognize those cells no matter what the image looks like, which is really important when it's looking at real patient samples.
That makes sense. It's like giving it a more well-rounded education.
Exactly. And they expanded their initial 180 images to over 1,800 using this data augmentation.
Wow, that's amazing. So they've created all these extra images without having to actually label thousands of new ones.
Exactly. And then they take all those images, those augmented images, and they split them into 3 groups. The training set, the validation set, and the testing set.
Okay, why do they need 3 different sets?
It's all about making sure the AI is learning properly and that we can accurately measure how well it's doing. The training set is the data that the AI actually learns from. It's like this textbook. The validation set is used during training to monitor how well the AI is doing on data it hasn't seen before, like a practice quiz.
Okay.
And then the testing set, that's like the final exam.
Yeah.
It's kept separate and only used at the very end to see how well the fully trained AI does on completely new data.
Wow, so they're really putting it through its paces. And they mentioned something called non-maximum suppression, or an NMS, with a threshold of 0.3. Can you explain what that's all about?
So imagine the AI is looking at an image and it finds a Ki-67 positive cell, right? It draws that bounding box around it.
Mm-hmm.
But sometimes it might actually draw several slightly overlapping boxes for the same cell.
Oh, I see.
And NMS is like this cleanup crew that comes in and gets rid of those extra boxes.
So it makes sure it's not counting the same cell multiple times.
Exactly. And with a threshold of 0.3, it means if those boxes overlap by more than 30%, the NMS algorithm keeps the one with the highest confidence score, which means the AI is most certain about that detection.
Oh, that's really smart. So it's like it's saying, okay, I'm pretty sure this is the right box, so let's get rid of the others.
Exactly. And it's all about making the final count of cells as accurate as possible.
Okay, so let's talk about why they specifically chose YOLOv8 for this task. The paper says it's much better for real-time object detection than the older types of AI, the convolutional neural networks, or CNNs. Why is that? What makes YOLOv8 so good for analyzing these really detailed pathology images?
So YOLOv8, it stands for You Only Look Once, version 8.
Oh, that's where the name comes from.
Yeah, and it's this really advanced system that's known for being both super fast and super accurate.
Okay.
Those older CNNs, they often have to process images in multiple steps, which makes them slower.
Mm-hmm.
But YOLOv8, it can look at the whole image in one go, which makes it much faster, hence the real-time aspect.
That makes sense. So it's like it's seeing the whole picture at once instead of having to piece it together.
Exactly. And that speed is super important when you're dealing with a ton of high-resolution medical images, like we have in pathology.
Yeah, totally. And they talked about some of the specific advantages of YOLOv8, like its anchor-free approach and the C2F module. Can you break those down for us?
Sure. So a lot of those older object detection models, they use these things called anchor boxes.
Ah.
Basically, they were like predefined templates of different shapes and sizes that the AI would try to match to the objects in the image.
Oh, okay.
But YOLOv8 doesn't need those templates. It figures out the bounding boxes directly from the image, which can make it a lot more accurate, especially when you're dealing with things like tumor cells that can have all sorts of weird shapes.
That makes sense. So it's more flexible and more adaptable.
Exactly. And then the C2F module, that's a special part of the YOLOv8 architecture that helps it learn and understand those complex visual patterns in the pathology images.
Oh, okay.
It makes the information flow more efficiently within the system, which leads to more accurate detection of the cells.
So it's like optimizing the way the AI thinks about the images?
Yeah, something like that.
And they mentioned a decoupled head for classification and regression and something called a feature pyramid network, or FPN. Those sound pretty complicated.
They are kind of technical terms, but they're really important for understanding why YOLOv8 is so good.
Okay, so break it down for us.
So a decoupled head means that the part of the AI that figures out what type of object it's seeing, is it a positive cell or a negative cell, that's separate from the part that figures out where the object is in the image.
Oh.
And this separation makes the whole learning process more efficient and more accurate.
So it's like having different teams working on different parts of the problem.
Exactly. And the feature pyramid network, the FPN, that's a clever way for the AI to learn from different scales within the image,
What do you mean by scales?
like different sizes.
Yeah.
You see those KI-67 positive nuclei, they can be big or small.
Oh, right.
And the FPN helps the AI detect them all, regardless of size.
So it's like giving the AI different magnifying glasses to look at the image?
Yeah, that's a great way to put it. And it makes sure that it doesn't miss any of those important cells.
And they even mentioned a bidirectional feature pyramid network, or BiFPN, which could be useful for images taken at different magnifications.
Yeah, that's like the next level of the FPN. It can handle even more variation in the images.
It seems like YOLOv8 is packed with all these really cool features that make it perfect for analyzing these super detailed medical images.
It really is. All those details in the images, the dense packing of cells, the variations in color and staining intensity, YOLOv8 can handle all of that.
And those techniques we talked about before, the non-maximum suppression, the confidence thresholding, the dynamic receptive field adaptation, they all help to reduce those false positive detections, right?
Exactly. They make sure that the AI is focusing on the right things, the actual KI-67 positive nuclei, and not just some random stuff in the background.
So they're like fine-tuning the AI to make it as accurate as possible? Okay, so they trained and tested 3 different versions of this YOLOv8 model, right? Nano, small, and medium. What's the difference between them?
Basically, it comes down to a trade-off between speed and accuracy.
Okay.
The nano version is the fastest, but it might not be as accurate as the larger models. The small version tries to strike a balance between the two.
Mm-hmm.
And then the medium version, that one is all about getting the highest accuracy, even if it takes a little longer to run.
And they use something called transfer learning, right? Where they fine-tuned models that were originally trained on this huge data set called COCO.
Right, COCO.
And COCO is all about everyday objects like cars and cats, right? So why start with a model that's been trained on that kind of stuff instead of just training it from scratch on breast cancer images?
Transfer learning is a really clever trick in AI. You see, those models that have been trained on huge data sets like COCO, they've already learned to recognize a ton of basic visual features like edges, shapes, textures, that kind of stuff. So instead of starting from scratch, we can take one of those pre-trained models and fine-tune it using our smaller, more specialized data set of breast cancer images.
So it's like giving the AI a head start.
Exactly. It doesn't have to learn everything from scratch. It can just build on what it already knows.
That makes a lot of sense. It's like someone who already knows how to drive, learning to drive a race car. They don't have to relearn the basics of driving all over again.
Perfect analogy.
Yeah.
So how do they measure how well these different YOLOv8 versions were doing? What metrics do they use?
Yeah, how did they know which one was the best?
Okay, so they use these standard metrics that are used all the time in object detection. First, there's precision, which tells you how many of the cells the AI identified as positive were actually positive.
So it's all about avoiding false positives.
Exactly. And then there's recall, which tells you how many of the actual positive cells the AI was able to correctly identify.
Okay, so that's about avoiding false negatives.
Precisely. And then they use this more comprehensive metric called mean average precision at 50 precision IOU, or MAP50 for short.
MAP50.
Yeah, and that takes into account both precision and recall and gives you an overall measure of how well the AI is doing.
So it's like the final grade.
Exactly.
Okay, so which one was the best? Which YOLOv8 model performed the best?
Well, the study found that the medium-sized YOLOv8 model consistently outperformed the others. It had an MAP50 score of over 85% for detecting those KI-67 positive cells.
85%, that's really good.
It is. It shows that the AI was really accurate at both identifying those crucial cells and figuring out where they were in the images.
That's awesome. And they noticed that the MAP50 score was higher for the positive cells than for the negative cells. Why was that?
Well, they think it might be because there were simply more positive cells in the training data.
Okay.
And also the positive cells, they stain this really distinct dark brown color,
Right.
which might make them easier for the AI to spot compared to those negative cells, which can have a weaker blue stain.
So it's easier to see the contrast.
Exactly. And the researchers also saw that the medium model just kept getting better and better at predicting those bounding boxes during training.
So it was learning effectively.
And it maintained those high levels of precision and recall throughout the training process.
So it was consistent too.
Very consistent. And because it had that perfect balance of precision and recall, it achieved the highest MAP50, making it the most promising candidate for actually being used in clinical practice.
Wow, that's really exciting.
Oh, it is. And there's actually a figure in the paper, figure 4, that shows you just how accurate the medium model was.
Oh, cool.
You can see the red boxes around the positive cells and the green boxes around the negative cells. It's really impressive how well it lines up with the original annotations from the expert.
So what's the big takeaway here? What does it all mean for the future of KI-67 assessment?
Well, I think the biggest impact could be the development of a much more efficient, scalable, and most importantly, objective way to measure KI-67 in breast cancer.
More objective, that's key.
Absolutely. This AI-driven approach could really help us get rid of that variability in KI-67 scoring, which we've talked about, is so critical for making the right treatment decisions.
Yeah, for sure. And the fact that they got such good results with a relatively small data set, that's really encouraging too.
Oh, it is. It suggests that you don't need millions and millions of images to train these AI models.
That's good news for the whole field of medical image analysis.
Absolutely. But there is one limitation right now: the technical expertise needed to actually use these AI models.
Oh, right. So you can't just install a program and be done with it. You need people who know how to work with AI.
Exactly. And the researchers acknowledge that in the paper. So what are the next steps?
Yeah, what's next for this technology?
Well, the researchers say we need to develop user-friendly tools that pathologists can use easily, even if they don't know a lot about AI programming.
So making it accessible to everyone.
Exactly. And they're also planning to expand their data set, including samples from lots of different hospitals.
Oh, that's smart.
That's important for making sure the AI works well on data from all sorts of different labs and patient populations.
It's not just tailored to one specific type of patient or one specific lab.
Precisely. The ultimate goal is to have this AI-driven KI-67 scoring become part of the everyday workflow in pathology labs all over the world.
Wow, that would be incredible.
It would. And it would mean more accurate diagnoses, more reliable results, and ultimately better care for patients.
So to wrap it all up, this research is really exciting. It's showing us that AI, and specifically this YOLOv8 model, has the potential to totally change the way we assess KI-67 in breast cancer.
It does.
And that could have huge implications for treatment decisions, leading to more targeted and effective therapies for patients.
And it's all about reducing that subjectivity, that guesswork that we've had to deal with in the past.
Absolutely. So as AI continues to become more and more integrated into medicine, it really makes you think, how is this going to change the roles of doctors and other healthcare professionals? And how do we make sure everyone has access to these cutting-edge technologies, regardless of where they live or their financial situation?
Such important questions to consider.
If you want to dive deeper into this topic, we highly encourage you to check out the research paper we've been discussing. It's a really fascinating look at the future of cancer care. Thanks for joining us for this deep dive.
It's been a pleasure.